Croptastic, the podcast by InnerPlant. We're continuing our series looking behind the scenes here at InnerPlant with the science team. Today we are joined by Ryan and Ian, who are part of the molecular biology team. Gentlemen, welcome. Let me have you introduce yourselves.
I am Ian Lemersal. I've been working at InnerPlant for about 1.5 years now, and this is my roughly third year since college working in molecular biology. As a molecular biologist, I always say I got it from my manager actually, but he always says something that I've really come to associate the job with, which is we're moving small liquids of small molecules into bigger and bigger tubes. And now ultimately we're working with think of biology, but fully experimental with a lot of things that you can't see, with DNA, proteins, other molecules that you can't see, and we're doing experiments with those things. I'm in the molecular pipeline, so I am working on a team with Ryan and Caitlin, and basically on the science team, and we're doing a lot of work right now with the genotyping pipelines, myself specifically with the genotyping pipelines, and with helping the transformation team assess and assay the plants that they've transformed.
And Ryan?
I'm Ryan Mitchell. I've been in InnerPlant for just under 2 years, and I lead our molecular bio platform. Before InnerPlant, I was at a couple different startups, one down in San Diego called Synthetic Genomics, mainly supporting projects around ag microbial discovery. And then I took a 5-year hiatus from ag and moved to the human microbiome, where I led molecular biology efforts for large-scale global clinical trials. So at InnerPlant, the molecular biology team is in charge of building constructs, so cloning regulatory elements out of plants, and helping build our biosensors, as well as genotyping our plants to make sure that the transformed plants actually contain our biosensors.
Walk me through the pipeline, how that process actually works.
Mm-hmm. Sure. So we our group specifically spans a couple different parts of the pipeline. So our cloning part is really at the start of it. So we take some of the regulatory elements or different DNA elements from plants or other sources and stitch them together with our biosensor design. And then once we have that construct, we can pass that off to our plant transformation group, and then they do the plant transformation, and then we come back and either assay the plants for presence absence of our biosensor, or in later generations we assay how many actual copies of that biosensor has been integrated into the plant genome.
So walk me through what you guys are working on right now.
So right now the biggest thing is, like Ryan was talking about, the assays with the plants that have already been transformed. Basically, right now we're mostly doing quantitative assays, so copy number assays, measuring how many copies of our construct are in those plants, those transformed plants. And right now the biggest thing is we have a pipeline going specifically for this copy number assay, where I'm extracting DNA from these plants that I'm getting every week at the start of the week, and then running the assay, collecting data, and then putting that data together in a certain way where the greenhouse team members and others will know what to do with those plants. And for the ones that don't have any of our tDNA, those are getting tossed. For the ones that are that are very strong hits, those ones are going to NETA for what we call our bulking pipeline. Those plants will be bulked, and for the ones where we're maybe not quite sure there was a hit, maybe it wasn't super strong, whatever, those will go to Arjun, and he's going to do further testing on those. So getting that pipeline going, having that be consistent, what we call the bulking pipeline. We're about a month and a half into that now, and so just having that be more consistent, just keeping that going for a long amount of time, and making sure that we're consistent with that, while also being able, at least for me personally, to be able to do some other stuff as well, and some troubleshooting on other things.
The purpose of that bulking pipeline is to allow us to have enough seed to go out into larger field trials, or do other work around pathogen assays or stuff like that. So it's all about seed availability.
Help me out, because you guys are kind of bookending the process, right? So you get the early squishing molecules together, giving them to the transformation team, getting plants back, and then breaking down the plants to make sure that the molecules are there. So at the very beginning of the process, the team comes to you and says, okay, we need a plant that does X.
Sure.
And then you guys go to work and just like find molecules for that?
Yeah, so there's a couple different approaches that we can take. We can find some different data sets, transcriptomic data sets that are out, been published in the literature, or we do in-house experiments to kind of see which genes are upregulated or downregulated during say a pathogen response. And then we can go after those pieces of DNA that basically say that the plant uses as like an on-off switch, and we can integrate those pieces into our biosensors that says like, okay, the plant has already evolved this piece of DNA that says when there's a pathogen, turn on. So all we can do is just take that piece of DNA, pop it into our biosensor construct, and hopefully see the response that we hope, which would be if there's a pathogen, our sensors turn on.
And it's going to be specific to what response we're looking for, like as far as what is the stimulant. So like, some research that Ryan's having me do right now is like around responses to insect attacks. So what genes are being upregulated during attack from pests, and that'll obviously come into play, as you know, like in our future research and things we're looking to do in the future with our sensors. But yeah, there's a lot of good stuff to be learned in literature, and that was something that definitely I learned a lot in like working in academia. Was there's a lot out there, but a lot of the challenge is finding the right stuff. But once you find it, it's very useful for what we're doing.
Now, earlier you were talking about having to build an assay. Like, walk me through like why you would need to do that.
So yeah, I mean it just depends what we're looking to do. For example, with the copy number assay, that was something that we wanted to do because it's tougher when it's something that's never been done exactly before. I think like obviously a copy number assay, qPCR, those things, like they've been done before many times, but in our specific context, we have specific needs. I mean, we're working with soy plants, for example, and there's just a lot of specifications that come from when you want to do something that hasn't precisely been done before. And so for that copy number assay, for example, we want to measure how many copies of our transgenic DNA are in those plants. And so we knew at least where to start to an extent, because we knew that qPCR would be the way to go for that. But designing primers and finding the right master mix to use and the right extraction protocol to get the type of DNA that we need, the amounts of DNA that we need, and to make it work, like those are all things that there's just so many little steps that go into it.
Right. That's crazy that because you're doing something new, you actually need to develop new tools in order to do it. And Ryan, help me out, because it's not just the assay, right? I mean, you guys, Ian was talking about the pipeline, and you guys kind of leading the department are kind of just like building a pipeline because this has never really been done before, and you're trying to scale it. So walk me through that kind of process.
Yeah. So our pipeline is basically, we do kind of a literature search, or we come up with new nominations of candidate genes that we want to go after. And then so the cloning team will clone those, make constructs, pass those off to transformation. Then they lead to Ian and then our plant phenotyping team that does kind of phenotypic characterizations of our transgenic plants. And so we are building plant-based biosensors. That's a really novel concept. So everything from around construct design needs to be novel. Our plant transformation team is doing novel work. Soybean is very hard to transform, and they are a rock star team and have made huge improvements just in general for soy transformation. And then our plant phenotyping team, they have to develop all these assays to say, do our sensors actually work? Can we see the signal? And so they have to be able to come up with assays and validate them to be able to allow us to then take their results, and allows it to kind of feed back into the construct design process, where we can then iterate upon constructs.
And there's a component in here that you're building. I know this because I'm cheating, because we have inter-team calls that I get to listen in on what you guys are doing. But you're building the process. Like, that's kind of an interesting thing. As you scale, you're adding team members. You're doing this. So you guys are actually building the process through which this would be done, because it's never been done before.
Yeah.
That's crazy. What's that like? Is that a unique challenge? Have you been through that before? Like, what's the deal with that?
I'd say every company has to come up with a process, and I think part of where InnerPlant is excelling is like how we're able to scale that. And so our constructs, we can crank through constructs, build lots of them at a time. And then you hit plant transformation where, at a normal company, they might only be doing, a couple constructs a month. Where at InnerPlant, we're doing, handfuls of constructs a week. And so that allows our phenotyping team to have to develop higher throughput assays because we're just making so many more transgenic plants. And so it's just, we're able to scale some of our assays to meet the demands that we're putting upon ourselves.
Got it.
Yeah, I mean, even from other teams too, everyone's doing stuff that is things they kind of have to be forward thinking with themselves because they haven't been done. Like even just the imaging, boxes that we use to look at the plants and to, measure and see signal. Even those have been made by, engineers that are on our team and all those guys. And it's just pretty much everything has to be a little bit, you take as much as you can from literature in our case, or from people here who've worked at other companies and know how certain processes or structures or organizations should work. Engineering people, they know how to make certain things, but everything's going to be a little new and specific for what we're trying to do. So
one of the things that I enjoy talking to the science team is from the outside, someone who doesn't do science and doesn't work in a lab. It seems lab's kind of quiet. There's not a lot going on. It seems like kind of a staid, stoic. But when I talk to you guys, you're all very passionate about what you do and you love what you do. Where does that come from? For you guys in particular, doing molecular biology, what do you love? What gets you out of bed to do that?
At least for me, I love kind of being just on the cutting edge of science, and through my career, I've kind of been on that edge, across different sectors. But at InnerPlant, we're doing something extremely novel, and that's what excites me about just pushing science forward.
Yeah, for me it's similar. It's mostly very similar, I'd say, which is that I just like learning new things. So I think that one thing that excites me, no matter what I'm doing, is just having the opportunity to learn. And when you're working in this field and at a company like this, and like I said, I've never worked with plants before specifically, and molecular biology is different for plants versus animals and all kinds of bacteria and all kinds of different things. It's so different. So working here in this field, but specifically here, is there's never going to be a shortage on things for me to be able to learn.
Well, what do you guys? There's a lot of interesting, exciting stuff coming up. We've got the Sentinels going out into the fields in Illinois. We have the Deer Syngenta deal going out. What do you? Is there something in particular you guys are looking forward to for the next couple months or year or whatever?
At least for me, it's around new trait development. So we're starting work on some new traits. So lots of discovery work and kind of seeing what's out there.
Yeah, I'd go with a similar answer because, having to, really start working on new sensor stuff, like with the insect pest sensor stuff that we're starting to look into, that's, again, another really good opportunity to learn. So, I'm getting to read some papers for that, and I'm going to probably be a little bit more involved as far as, being able to learn about everything that's going on every step of the way. And now that I know how a lot of this stuff works, and specifically with the pathogen stuff and the sensors we've already built, now that I understand kind of how that works, getting to apply that to something new like pests, for example, is it's a totally different thing, but it'll be in the same mold as kind of how we've already done things now that we have that structure. So it'll be really interesting. A lot of good opportunities to learn, coming up here with new sensors and things being worked on, and even new types of crops a little bit. I was doing some work with corn the other day, which I haven't done before. It's mostly been soy and a little bit of tomato for me, but just getting to continue to expand.
That's great.
Yeah.
All right. Well, thank you guys so much for joining. This was Ian and Ryan from the molecular biology team continuing our look behind the scenes at InnerPlant. If you have any questions, leave them in the comments below. Feel free to reach out via Twitter or on LinkedIn. And thanks for watching. We'll see you next time.